So Hiya, are you there?
Yeah. Oh, sorry, I was on mute.
Okay. So on behalf of NIPER-Ahmedabad, I welcome all of you to the third day of the lecture series. So today we have another two excellent speakers, Dr. Hiya Ghosh from National Center for Biological Sciences, Bengaluru, and Dr. Rajneesh Chaturbaidi from Indian Institute of Toxicological Research, Lucknow. So we will start with Dr. Hiya Ghosh. Before starting, I would like to introduce her to all of you. So Dr. Hiya Ghosh received her PhD degree from University of Pittsburgh and postdoctoral training from Columbia University in the United States. Her investigations into adult blood cells using in vivo fate tracking was among the first few evidences that demonstrated a requirement for active maintenance of adult mature cells. She was awarded the prestigious Scholars Award from the American Society of Hematology for her investigations during her postdoctoral training in immunology. Before starting her laboratory in NCBS, she worked as a faculty in the Department of Neuroscience at Albert Einstein College of Medicine, New York, studying brain regeneration. Her laboratory at NCBS focuses on adult brain homeostasis and neuroinflammation, seeking to understand genetic regulations underlying maintenance in the adult brain cells and deregulations thereof that may contribute to neuropathologies. She has received the Ramanujan Fellowship and is currently an EMBO Global Investigator. So today we are going to hear, she's going to talk on maintaining form and function of cells in the adult brain. So over to Hiya.
So first of all, thanks, Pallav, for organizing this. I'm absolutely delighted to be part of this program. Also thanks for the introduction. A small correction there. I was in Albert Einstein, a fellow, not a faculty. Also, I would like to apologize right in the beginning that I have a grant progress report meeting to present, abruptly sent to me to do that at 4:45 today, so I'll have to rush. So real apologies to Dr. Chaturvedi. I would have really loved to listen to him too, but I hope you can see the constraints here. So without further things, so we can spend some time in question answers also. I'll just jump directly into what I plan to discuss today. So like how Pallav said, that my lab is really about understanding how homeostatic processes act to maintain the normal functionality of the brain. And so to do so, first thing to pay attention to is that the brain consists of many different types of cells. So of course there is neurons, everybody knows, but also we know that there are astrocytes, there are oligodendrocytes, microglia, and also there are adult neural stem cells, right? So after they develop, and we are talking about the adult brain, how are they maintained throughout the lifespan? And this is kind of a little bit of take home from what I worked in my postdoctoral studies, but in a different system, the hematopoietic system, where the work showed that after a cell is mature and is differentiated, it's not a done deal. It's not a permanent verdict, and that active maintenance is required. Otherwise, it may not perish, but it may become another cell type. It may do other things, right? So on that same concept, what we're trying to now apply is that, does that apply to other organs, including in brain? So the work in my lab is focused in brain, and here, like I said, the word maintenance is essentially alluding to the fact that, are the cells doing what they are intended to do? And if so, are there active regulations that allow them to be able to do the intended task, right? And this has a word in biology, we call it homeostasis, which is the optimal functioning of an organ or the steady state. And we know that homeostasis is actually regulated through very intricate genetic regulation. And in the context of brain, we want to understand what are those actions and do so. And here the motivation is that if one is able to understand what it takes to be normal, then perhaps you can decode pathology better, because it would be some kind of deviation from normal. So defining normal is kind of important. Now, in the scope of what my lab is trying to do is to understand it in a cell-specific manner. And why so? Is because the maintenance or the regulations that one would think is required for the cells to perform and be what they're supposed to be could mean different things for different cells. And why so? So just very intuitively, if you think about neuron, it is a cell whose main characteristic is to be physically well connected through the dendritic tree that it has, which allows it to kind of transmit the information that it's supposed to be, right? So you would imagine that maintenance for it would involve stability of the dendritic structure throughout a lifespan, just one aspect of the maintenance, right? However, now if you consider this criteria for neurons to be maintained, there is another cell in the brain, microglia, for which maintenance means completely different thing. This is the glial cells in the brain with immune functions, right? And I'll get to a little bit more to what this means, glia and immune function, but essentially it requires different structure to perform the different function. So, right, the dynamic transformation of doing different things and becoming different things is kind of integral to its homeostasis. The homeostasis of microglia depends upon becoming something else, performing a function, coming back to normal to do the normal function that it does, right? So the context is very different, even though it is still maintenance. And now you have the third cell type, which is the neural stem cell in the adult brain. And by definition, it is a cell-making cell, right? So this is a cell-making cell, and what intuitively comes is what it can make or what it should not make would be integral to its normalcy. So it would do its normal function only if the regulation or the program that it gets to make another cell type is well defined and is adhered to, right? So in the context of adult neural stem cells, the context is different. Fate restriction and fate potential is important. So now in this context, what we strive to understand, just taking these three cells, the neuron, microglia, and the neural stem cell, we want to figure it out. How are the programs? Do we know all about it? Or if we perturb it away from homeostasis, can we learn more about what all it's capable of doing? Yeah, so in this context, I'll start with microglia first. And microglia, this is a nice art that my student Vinaya made to kind of describe the dynamic work of microglia. So here in the blue, you have a homeostatic representation of microglia looking calm, having thin dendrites around it. And on the right-hand side, you have an activated form of microglia. You can see phagocytic cups and sort of imagine like an angry microglia, right? This is just figuratively. But really what I mean to say is that in a healthy brain, it does different function. And just as an example, one of its function in the healthy brain is to enable or help synapses, right? And that includes both spine formation and elimination. Spine are the functional unit on the dendrites of a neuron, and they are continually being produced or not based on the transmission that has to happen. And this is enabled and helped by microglia. On the other hand, if you look at its immune role, that is actually not the steady state role. It is the role that is invoked when there is disturbance from the physiological conditions, such as infection, injury, disease, any of it. Then it has to perform things that are very deviant from what it does normally. It has to make cytokines, chemokines. It also is capable of antigen presenting, a whole different cellular machinery you would imagine that is needed, but performed by the same exact cell that resides in the brain, right? So you can imagine that the program, genetic program that will allow you to perform this or that would have to be substantially modulated based on the task at hand. And so then the question is, can all microglia, we know there are many, many microglia in the brain, can all of them do all of these functions? Or are there subsets? We know in the immune system, for example, T cells is not just one cell type. There are many variety of T cells. However, we don't know of such a thing for microglia in the brain. Is it possible that they have subsets, or is it that each of them can do all the things that they are required to do if called to task? And the other question to it, related to it, is, so it's a very common phenomenon known in all neuropathologies that there will be activation of microglia. If brain is not feeling good, microglia will show up in activated state. And if so, then, a very important thing one can imagine for normalcy to come back or homeostasis to come back is for the microglia to get deactivated. right? Because it has different function in the deactivated form. So the question is, do microglia revert back to deactivation or the homeostatic state, or are they replaced? The activated one dies and a new one comes to kind of make up for it. So these are burning questions in the field, and we kind of try to also pitch in our understanding into it. So to understand this, we created an injury model, a relatively very nominal injury. What you do is you basically poke a needle into the brain of the mouse. You don't even drill, you just put the needle on across the skull and do that on one side, so the other side is contralateral control, and you see immediately that there is huge amount of microglia that you can see compared to the normal state, meaning microglia has proliferated, a process known as microgliosis. You can also see proliferating cells, Ki67 positive, and you can see that the injury site is very clearly visible. And now, so now the question is, you have perturbed the condition and you've allowed the microglia to become activated. And now can we use this system to understand what goes on? Is there replacement happens? But even before that, what really goes on inside that at the cell-to-cell level? So to do that, we performed single cell transcriptomic. This is a very, as you all know, a very powerful tool that we have available to us these days, where we can, understand 25 or at least 8 to 10,000 different dimensions of a cell by looking at the expression of these genes, right? So here what we did is we took this little area where injury had happened and correspondingly a control area, and we sorted microglia first. So we are basically focusing on what's going on with microglia, although we know that at injury site, the niche cells, all the other cells are also doing something. But for this study, it's focused on microglia. We sort the microglia out and we do single cell transcriptomics. What we see readily is that there are different clustering, meaning there are different population in the site of microglia in the site. So this is uninjured. So this is the contralateral side. In normal healthy brain also, you see different clusters, populations of microglia that cluster away from each other. And very noticeably, there are two predominant clusters. We call them homeostatic by matching their gene expression. And then we see minor populations, 3 of them, right? And now when we compare this situation of steady state with the situation where we have evoked activation by injury, what we see is very interesting. We see that the homeostatic microglia largely remain the same. Yeah, so you can see that cluster 2 and 0 are largely same. What in fact happens is that this minor population, which anyways cluster away from the homeostatic microglia, is the one that expands. And then we have a very big cluster 3, and given the size of it, one would think that it's probably important for this response. And this is day 3 of injury. So there is some interesting data more that we have on it to understand what might be going on. But at this stage, and since it's not solidified yet, I thought we'll talk about the other stories. But what we understood is that minor population 1 and 2 are to do with immune response and proliferation. However, this population that shows up as a big one seems to be deviant from these 2 populations and may have a role to play in maintaining these 2 homeostatic microglia at the site of injury. So this is very interesting. And to summarize what we find so far is that when injury happens, when you perturb the physiological situation, we see that there are populations that already existed, but in a very scanty quantity. Those are the ones that proliferate. And somehow there is huge amount of normal microglia that are still present and sustain themselves in the state of injury. And so this is very important for us to know what these new populations or expanded population might play a role in maintaining the homeostatic population at the site of injury. And then in due course of time, to find out how all of this plays to ultimately regain homeostasis, right? So, so that whatever has tissue injury or whatever else has gone wrong, how do you bring it back to the steady state? So we are currently working on understanding what is the functional relevance of each of these subsets that come up, what could be their regulators, and how might they play a role in tissue reinstatement after injury response. So stay tuned, and I hope we'll get some answers pretty soon. So now switching gear to the other cell type, which is the neural stem cell in the adult brain. And so I see a question in the chat box. So can I take the questions at the end of the talk, perhaps? Is that okay? Just to keep the flow going?
Yes, ma'am.
All right, great. So, so the other cell that I'm going to discuss today is the neural stem cells in the adult brain. And we know now for several decades that adult brain actually harbors neural stem cells, which is very exciting because neural stem cells can make neurons and glia. However, there are some, it's important to know the limitation or the extent to which we can be excited about it. Yeah. So first thing is that it's not there all over in the brain. This is a mouse brain, and this is my cursor visible? Is my cursor visible? Can somebody tell me? Perhaps.
Yes, ma'am.
Right. Yeah, okay, all right, great, thank you. So, so this is a mouse brain and this is a human brain. And the green area is the only area where newborn neurons have been identified from postmortem samples or from rodent mice, right? And you can see how minor this area is compared to the total brain. So that is the extent of where neural stem cells are there in the brain and how much new neurons can be is seen in an adult brain. And this is a immunofluorescence image of a mouse brain where only the stem cells are genetically marked with GFP. So you can see this thin line. So essentially, this is telling you the quantity and location of the stem cells in the adult brain. So you can see how sparse it is. It's really along the lateral ventricles, the line of the lateral ventricle, and then a little bit in the hippocampus in the dentate gyrus area. And if you zoom it, you can see this entire blue thing is the dentate gyrus with thickly packed granule neurons in them. But the green cells, which are the stem cells, are really only the first 2-3 cell layers at the very start of the dentate blade. So that's the amount and scarcity of cells. However, even if they're very low in number, they do very important functions. And these have been mainly found through rodent experiments in rodents and other species. And the consensus is that the newborn neurons that are formed in the adult brain play a role in selective type of cognition. And one of them is pattern separation. This just to give you an example, you can see 2 black creatures on the slide, and I'm pretty sure that most of all, if not all of you can distinguish that one is a panther and the other is a cat. And that is because we have the ability to code in detail what we are seeing and yet keep them orthogonally separated so that the overlap of information does not happen, mixing up of information does not happen, so that we can pattern separate even if the two things are very, very similar to each other. So this is the cognition where newborn neurons have been shown to play a role. The other and related one is a reversal learning, where again, in a way, our survival greatly depends in our ability to forget and relearn, right? And an example will be, let's say you had a bad experience while walking on a trail, it was gravelly and was in slope and you slipped a lot of times, and you associate that context with that fear or that unpleasant experience, that can make you, unable to do this again ever, unless you're able to go on a second hike, maybe in the same place with better shoes, and recode the same context with a better experience, right? It's just an example to show how important reversal learning is for us to strive and survive. So that is another cognition where new neurons are known to play a role. And finally, and I think very contextual to our lifestyle these days, is our ability to be resilient to stress and anxiety. Right, and these stress and anxiety related data has been largely correlated, but more important studies, mechanistic studies are coming the last 2 years. So it seems like it definitely plays a role. But the good news here is that these stem cells that are scanty in amount and make a certain type of neuron doing a certain type of participating in a certain type of cognition, actually respond to the environment of the organism. So this is very important thing about the neural stem cell. So this has been shown through rodent experiments, where this, on the left hand side, you see a normal cage where there is bedding and the mice are there. On the right hand side, EE stand for enriched environment, and you can see there are tubes and wheels, and the mice can go around and quite voluntarily choose what to do, but there is heterogeneity in their environment to kind of play around, and therefore enriching for their experience. Now, if you go look at the number of newborn neurons marked by these white cells marked with double quoted, you can see that the standard case has a certain number of new neurons, which is dramatically increased if the same mice were in enriched condition, and was dramatically decreased, on the other hand, if they underwent stress. So social isolation is a form of stress to mice as it is to human. And you can see there is a direct impact of how the neural stem cell is behaving. It's not able to make enough neurons, right? So in all, what we know about stem cells is that it is able to make neurons, and these neurons can influence both cognition and mood. And on top of that, these stem cells can be influenced through very, lifestyle changes, non-invasive things, changes that can influence these neurons, which can in turn influence mood and cognition. So definitely it's interesting and probably useful to learn more about what these stem cells are, what all they can do, and how they do what they do. So what do we know about stem cells? Again, fate decision is important because it is a stem cell, and what is known already. So this is how the stem cell looks like. It's called a radial glia-like cell because it has a radiating process, a singular radiating process differentiating it from neurons, and it's called RGL. Therefore, this can make astrocytes. It can self-renew itself, make 2 copies of them, or it can differentiate into neuron via the intermediate proliferating cell, which is a neural progenitor, and then go on to become a neuron. But the question is that all that it can do, right? Or are there more things that are tucked inside and we don't know unless we perturb and find, we can perhaps find more. And so for doing that, we actually went about this transcription factor called TCF4. And this little frame is coming from this dentate gyrus. Let's say a little frame from this dentate gyrus. You can see a lot of granule neurons thickly packed, all these DAPI positive cells. And you can see the first 1 or 2 cell layers that are marked with nestin GFP, telling that these are the stem cells and their progeny. And you can see that TCF4, while this is also expressed in the neurons, it's very nicely expressed in all cells green at a very good quantity. So what is TCF4? TCF4 is not only a transcription factor, but it has relevance to the brain. It's very important for brain development, is already known to be important for neural stem cell differentiation and migration during embryonic development. Yeah, and it's also implicated in psychiatric and neurodevelopmental disorders, including schizophrenia. But the task we wanted, so if we want to know what TCF4 is doing in neural stem cells in the adult brain, the challenge was that how do we know it? If we delete, if we do a loss of function, then all these neurons are going to be affected, and we wouldn't know what was the cause, what was the consequence. So it was important to be able to delete in the live mouse, but only do so in the stem cells. And this is totally entirely possible because of the cool mouse genetic tools that are available. You can use the nestin promoter to express a Cre recombinase, and by virtue of that, when you induce the recombinase, the deletion will only happen in the green cells. And at the same time, you can mark them green by also expressing a GFP protein through the similar mechanism, and therefore anything that happens to this cell, you can trace it by tracing the green cell. And what we see is that immediately after TCF4 deletion in the neural stem cells, there is a decrease in the number of newborn neurons as quantified by DCX positive cells in the adult brain. So what happened to the progenitors that they could not make neurons anymore? And again, to understand this, we did a lineage tracking, genetic lineage tracking, because why this technique is important is that it allows you to, inside the brain, track for what is happening to a cell that has been genetically altered, right? Because you induce the green color while altering the gene. And now, even if it stops expressing the neural stem cell protein or the neuron protein, you can just find what this green cells is by testing many other colors and co-localizing it with green. So we did this, we used this strategy and we induced deletion and making the cells green in wild type and knockout mice in an adult stage, and then went on looking at various time frames to see what is happening to the green cell. And, consistent with what we had seen before, that fewer of the green cells actually became DCX. But if you look at the timeline, we see that it actually is affected at the proliferation stage, right? So if I could go back to this schematic, which I did not explain earlier, is that the neural stem cell has to go through this intermediate transiently amplifying progenitor stage before it can become a neuroblast and then go on to become a neuron. So what we saw through our lineage tracing is that, yes, definitely DCX was reduced, and therefore mature neurons, newborn neurons were also reduced, but the cause was here, the proliferation had reduced. So the next, and the other thing also we found out is that this decrease was not happening due to neural stem cells becoming something else. There was no astrocytic fate going on here, but indeed it was proliferation. And as I told you, these cells are very, very few. When you take out a brain and want to extract neural stem cells, they are prohibitively low in number. So what we had to do is we had to take the cells and culture them. And very nicely, these cells grow as neurospheres with this tight border, nicely clonally expanded tight cells. After you grow them, if you now delete DCF4 in them using the same lock-speed-3R mechanism, what you see is dramatic differences. You see smaller neurospheres, but not just that. You also see different morphology. It's all spread out and not quite neural stem cell looking like. This is the number quantitation to show that there is, they cannot make neurospheres. But what did they become? So you do a secondary neurosphere culture to see is the stemness preserved. If you took these cells and allow them to make another neurosphere by clonally expanding, would they do so? So we did the neurosphere culture and we saw that TCF4 deletion was killing their stemness. They could not make self-renew themselves. So what else? They cannot self-renew, but what happened that they cannot self-renew? To get an easy answer, you can just look at the transcriptome. Because TCF4 is a transcription factor which affects gene regulation, you want to know what did it do, what went up and down that this became. And immediately enough, we saw that there was an upregulation of these genes, which to somebody who knows a little bit myeloid biology will all look like, not like neuroectodermal or neuronal or glial kind of cells, but it was very myeloid looking like signatures had come up. And of course, we did ontology to this collection of genes, which showed that there was indeed, these activities, inflammatory response, cell adhesion, other kind of cytokine production going on, which were indicative of inflammation or myeloid kind of response. And then to confirm that, we took this set of genes and we compared it with the bona fide set of myeloid differentiation kind of gene, and we saw that yes, indeed, that entire gene set was upregulated in neural stem cells that were knockout for TCF4. So if that is so, that they are transforming or losing their neural stem cell property, then we would expect that they will not be able to differentiate into neurons. And so we checked that. We differentiated these neural stem cells to become neuron. And in the upper panel, you can see that they very faithfully make these TUJ1 positive neurons. The other confounding genes are completely not expressed. They're making thin, nice processes as would be expected for neuronal differentiation. But in the case of the knockout, first you see fewer progenitors, and whatever survive after TCF4 deletion, you see that they make funny things. These are stocky, bushy kind of cells, which also express MBP. This was one of the upregulated genes we saw in our transcriptional profile. And so alongside TUJ1, they also express MBP, which is a oligodendrocyte marker. But not just that, we also saw that they also express CD68, a myeloid gene marker expressed only by microglia in the brain. So clearly, as a result of TCF4 deletion, these stem cells went confused and could not faithfully make neurons. So other than that, other than their ability to lose their ability to become neurons, what would one expect is that since there was myeloid mark, such enhanced mark of myeloid transformation, there should be inflammation, one would expect, or we wanted to test how far could this be true. So to do that, what we did is dissected the entire dentate gyrus, which is the niche of hippocampal neurogenesis. And we tried to look at the signature. And we saw that in the knockout brains, where the only alteration was TCF4 deletion in the stem cells, there was heightened mark of inflammation, interferon gamma, interferon beta, everything that would suggest that inflammation is going on in the niche. And as we all know, microglia activation is a hallmark of inflammation. So we also checked as a secondary response, are microglia activated through checking upregulation of CD68, a lysosomal marker, and indeed that was also visible. So in all, what we found is that the adult neural stem cells, which was known and is understood to be only good news, making capable of making neurons and glia, actually harbor a inflammatory potential, right? And in the at the upfront, one may say that this could be a detrimental potential, but we do not know about it. All we have been able to find out is that there is a possibility or a genetic program that can transform these cells into inflammatory. But why would this potential be? What is its physiological relevance? Inflammation, although the sound of it is not really happy sounding, but inflammation is actually a protective mechanism. It's used many a times as a protective mechanism towards homeostasis for the physiology, by the physiology. And so we wonder if this potential has some role to play in physiologically perturbed condition. And in the same vein, is it possible that if it is dysregulated or this aberrant expression of this latent potential, could it play a role as a contributor to various inflammatory neuroinflammatory diseases, including neurodegeneration? So these are some of the research interests that are on progress in the lab, and hopefully some other day we can talk with some exciting data. So finally, the last segment is kept for the star cell of the brain, the neuron, right? And as I said, the structure of the neuron is kind of very, very important for its integrity throughout the lifespan. And of course, it contacts each other. This is how the contacts is what it allows it to kind of do what it's meant to do, which is transmit information. And so question is, once this, and we know that these neural circuits are laid out during embryonic development, are functionalized in the postnatal critical window period, and then after that, it just stays. It stays stable. I'm talking about the dendritic structure, not the spines. It stays stable because it's required to be stayed. The question then is there a proactive mechanism? How is it ensured? Because lifespan is long in many species, such as in us, it's decades, 7, 8, 9, 10 decades. How is it ensured that this integrity happens? And it's not naive to be asking this, because there are instances, two types. One is in pathology, where you see atrophy, structures becoming either hyper or hypo atrophied. The other example is during, very few of these examples are there, where during structural plasticity in the adult brain has been correlated to a functional outcome, right? So it is also possible that these dendrites, while they are needed to be stable, can also be utilized to alter connections, or perhaps use the structure to change different functions. of neurons. So the question here is, how much do we know about the structural changes and the regulation behind that? So in this study also, the same transcription factor kind of help came in very handy for us. Earlier, I just showed you how it looks in the hippocampus in the dentate gyrus area. You can see here the dark magenta, bright magenta color. And if you now zoom out and look at the entire parenchyma, what you see is that there are dots all over the brain, indicating DAPI is the cell nucleus and TCF4 in red is green. So every pink dot is a cell expressing TCF4. So it's quite impressive to see how widely and abundantly TCF4 is expressed in the adult brain. And we then asked, and obviously that said that a lot of neurons expressed it. So we wanted to go methodical about it. And we looked into different neurons, both in excitatory and in inhibitory class in different regions of the brain. And here CAM kinase 2 alpha is marking excitatory neurons. You're looking at the cortex, and you can see all these positive for excitatory neurons are also positive for TCF4. And in the same place, now if you use the inhibitory neuron marker, you see TCF4 expressed also in the inhibitory neurons of the cortex. And this was not only true in the cortex, we saw the similar pattern in hippocampus, also in striatum, and interestingly also in the cerebellum. So the forebrain, the cerebellum, you see TCF4 being expressed quite pan-neuronally. But I wouldn't say pan-neuronally because we have seen that there is some dynamic to it. So let's say it's very widely and largely expressed and is not restricted to a neuron being inhibitory or excitatory. So obviously, and given that it's steadily expressed throughout a lifespan, one would wonder that this transcription factor, which is known to be neuronal development, it did its job during embryonic development. So it still continues to express throughout a lifespan. What must it be needed for? So simply on that hypothesis, we went about the same strategy using this cool tool of inducible deletion, which allows us to delete TCF4 only from excitatory neurons, right? And do so only in adult animals. So the confound that could have come from developmental defects due to absence of TCF4 was not there. This was a mouse that was grown well, completely wild type, everything fine going on in the brain. And then you go one fine day, delete TCF4 from only the excitatory neuron. What do you see? Immediately, you see a huge structural change. This is within 2 to 3 weeks. You see more dendritic arborization. And you also see that the activity that looks like these are action potential spikes. You see it at a certain interval and amplitude here. You see while the amplitude is more or less same, the frequency changes a great deal. So it becomes hyper excitable. So clearly, the continued expression of TCF4 in mature neuron is definitely needed for the neuron to maintain not only its structure, but also its electrical excitability, the baseline electrical excitability. So the question is, what is it doing? Because if we know it's a transcription factor, it's definitely regulating it through, altering gene expression. If we know what it takes for a neuron to maintain its structure and function, we learn about what it takes for it to be normal, right? So we want to know what TCF4 is doing. And in doing so, there was a little bit of challenge, and this is just to show you how we go about this. Here, the green cells that you're seeing are excitatory neurons in which we deleted TCF4, but at the same time, we mark them green by expressing GFP. So only in neurons where TCF4 is deleted or CRE is activated are the neurons that will turn green. Now, if you take a close look in this region, you can see even though all of these neurons are CA1 neurons, pyramidal neurons of the hippocampus, not all of them are green. Not all of them, that means, have deleted TCF4. And since they are so closely packed, enough number of negative and enough number of positive neurons to have TCF4 or not, if you took this microdissected this area and did any kind of analysis, your results will be there will be a high noise in it. So the task here was to be able to pick out only the green cells. Yeah, so to be able to understand what went wrong when you deleted TCF4 from them. And this was pretty tough because it's not blood cells. These are entangled neurons of an adult brain. Anytime you're processing cells from an adult brain, it's a nightmare because neurons are very, very sensitive. They don't live very well if they're taken out and kind of processed with. But we standardized our procedure, and we actually used FACS, a technique that is used in the immune system so much more. We used that to extract adult neurons, the green ones, in which we had genetically altered TCF4 and seen those differences. And this effort was well worth it, because we only got a list of 100 genes that we found was differentially regulated, and majority of these were upregulated, meaning TCF4 had a suppressive role in neurons. So that immediately tells you that the proactive maintenance of the neuron depends on suppression of certain gene signaling, gene network, in absence of which there is changes in the structure, in the excitability, both. And when we looked deeper, we saw that a larger fraction of these upregulated genes actually belonged to, microfilament and ciliary kind of processes. So this was kind of unexpected, but very exciting for us now. So again, to summarize, what we saw is that even in the case of neurons, as we found in the case of stem cell, proactive maintenance seems like to be important and meticulously regulated through continual expression of TCF4. And other than that, what we come to learn is that there is this ability in neurons, mature neurons that are already connected in a complicated circuit, there is latent ability to change their structure. So we are now very intrigued towards two questions. One is that why is this ability maintained? It would be so much easier if it was a done deal and the structure was there, stable forever, but it's not so. And proactive suppression, why that is, and could it be possible that this ability to kind of morph itself differently is preserved intentionally so that it can be used as an adaptive response, let's say in a situation when you lose a few neurons, right? So these are interesting ideas that we are now trying to get into to understand. And in that, the other affiliation that came out from it is the involvement, the possible involvement of cilia-like processes, ciliary processes in kind of sensing perhaps the environment, and therefore being part of this process of possible flexibility of structural change. So we are looking into these two aspects. So to overall, I think the take home from what we have understood about through our investigations is that active maintenance is integral, even if it is adult brain, even if it is cells that are laid out during, the embryonic development and everything, it still depends on active maintenance. And in the case of stem cell, you see that otherwise they have tendencies to transform into inflammatory. In the case of neurons, you see they can change their structure and possibly connections. You also learn that there are latent possibilities that we do not come to know if we are just looking at normal physiology. It is only through perturbation that we come to know about latent potentials. And if we are able to know all the potential that is there and all the regulations that are required for normalcy, then perhaps we would be able to better use them for, every translational use as well. So these are the people, my lab, who have, a great bunch of people, all the funding agency and CBSTIFR, DST, DBT, and EMBO. And, it's such a relief that COVID is coming, hopefully, to a pause, and we had to celebrate with Rasgulla and Fuchka. So these were brought from Calcutta for my lab people to kind of mark that we can eat together now, sort of. All right. So with this, I think I'm ready to take any questions anyone may have.
Thank you, Hiya. Thank you for the wonderful talk. I open the floor for the question. If the students have any questions for you. So is there any question? So I have a question in the chat box here.
It's disappeared. Or is there? Okay, fine, great.
So I'm going to read it out.
Sure.
So for the single cell microglial population, does your lab use magnetic beats or FACS to identify microglial population? Or
No,
you utilize any
FACS.
other methods?
We do FACS.
You do FACS, okay.
We do FACS.
So here I know you are in rush, so no more questions. So thank you very much for your time. That is more precious for us. And hopefully we will like you to be in person at NIPER-Ahmedabad once we are out of this pandemic. And I welcome you in for any future conference or anything at NIPER-Ahmedabad. So do visit us and interact with our faculty members, and that we can grow well and we can collaborate with each other. Thank you, Hiya. Thank you very much.
Thank you everyone for your time, and I would love to come back in person sometime.
So I would like to invite Dr. Hemant to introduce our next speaker, Dr. Rajneesh Chaturbaidi from IITR. Dr. Hemant, please.
So thank you, Dr. Ghosh, for your exciting talk. So today we have another excellent speaker, Dr. Rajneesh Chaturbaidi. So he did his PhD from IIT, that is IITR, Lucknow, and also he finished his postdoctoral at Cornell University. Currently, he is working on different neurodegenerative diseases, including Alzheimer disease, Parkinson disease. His main aim is to find out how environmental toxin affect or modulate the disease pathology. So basically, he have published a lot of paper in prestigious journal like ACS Nano, Nature Medicine, JBC, that all are very reputed. And today he is going to deliver a talk and on the topic regenerative medicine in Alzheimer disease. So with no I will just welcome Dr. Rajneesh, and please feel free to ask any question in between or put your question in the chat box. Welcome, Dr. Rajneesh. I think you are on mute. Please unmute yourself.
Can you see my slide now?
Yes, we can. We can see your slides. Just make it full screen. Yeah.
Okay. First of all, thank you so much, Dr. Pallav and NIPER-Ahmedabad for giving me this opportunity to present my research work in front of you all. So I would also like to thank for brief introduction. So today I am going to speak about regenerative medicine in Alzheimer disease. So Dr. Hiya Ghosh has talked a lot about neural stem cells. She has given very brief introduction about the neural stem cells. So she has made my task a little bit easy. So today I will be talking about regenerative medicine in Alzheimer disease. Since I can see that lots of students are here in audience, so I will give a very brief introduction about neurodegeneration. Neurodegenerative diseases are the diseases of progressive loss of structure and function of neurons, and it can be due to degeneration of specific type of neurons. Like in the brain, we have several types of neurons like dopaminergic neurons, cholinergic neurons, GABAergic neurons. So the degeneration of specific type of neurons leads to onset of specific neurodegenerative diseases. In onset of neurodegenerative diseases, several factors are involved, like genetic predisposition plays an important role. We know that 90% cases of neurodegenerative diseases are sporadic in nature. The etiology of those factors are still not known, but it is believed that different environmental factors plays an important role in pathogenesis of sporadic neurodegenerative diseases. Similarly, genetic predisposition also plays important role in around 8 to 10% neurodegenerative diseases. So genetic predisposition involve mutation in different kind of genes. And similarly, oxidative stress, mitochondrial dysfunction, impairment in autophagy, mitophagy process. So these kind of factors also plays an important role in pathogenesis of neurodegenerative diseases. And then aging is a normal phenomena by which neurodegeneration takes place. So on the basis of degeneration of specific types of neurons, there are several diseases. Alzheimer disease is called somebody has some question?
No, it was like someone was on mute. Like, sorry for disturbance.
It's okay. Yeah, so Alzheimer's disease involves degeneration of cholinergic neurons, while in case of Parkinson's disease, there is degeneration of dopaminergic neurons. And in case of Huntington disease, there is degeneration of motor neurons. And in amyotrophic lateral sclerosis, also degeneration of spinal cord and motor neurons takes place. So my group is basically working on Parkinson's disease and Alzheimer's disease. So today I will be mainly focusing my talk on Alzheimer's disease, how we can enhance brain self-repair mechanism in Alzheimer's disease. So what is Alzheimer's disease? It is a chronic and progressive disease which characterized by progressive loss of cholinergic neurons into the cortex and hippocampal region of the brain. And the main symptoms of these diseases involve learning and memory deficits. So in advanced cases of Alzheimer's disease, patient, they cannot learn or memorize anything. So these are the main symptoms: memory loss, impaired decision making, and then they also have language problems. So here is the schematic diagram which suggests that the pathogenically that in the brain of Alzheimer's disease patient, they have amyloid beta accumulation, which is known as senile plaque. And then due to these accumulation of amyloid beta, the healthy neurons, they die. So here you can see this is the healthy brain, and this is right hand side, it is the Alzheimer's disease brain. And Alzheimer's disease is characterized by shrinking of the different brain regions like hippocampus region and then the cerebral cortex. And in Alzheimer's disease patient, there is accumulation of extracellular accumulation of amyloid beta plaques and then intracellular accumulation of neurofibrillary tangles. So the mechanism of neurodegeneration mainly involve accumulation of beta amyloid and then hyperphosphorylation of tau. Tau is a protein which is mainly involved in this synapse formation and then microtubule stabilization. In case of Alzheimer's disease, hyperphosphorylation of tau protein takes place, which results in degeneration of microtubules. So several therapeutic approaches are being used worldwide to inhibit neurodegeneration through inhibiting the tau hyperphosphorylation. And similarly, the approaches are also being made to stimulate the regeneration of brain's self-plasticity approach. And whatever the drugs are available in the market, they only provide the symptomatic relief. They cannot halt or disease progression. So this is the major limiting factor with Alzheimer's disease therapeutics, that whatever drugs are available in the market, they cannot halt the progression of the disease. They just provide the symptomatic relief. So for the sake of student, this is the healthy neurons, and these healthy neurons, they make connection among themselves, and then they do the neurotransmission through releasing the neurotransmitter, and signal passes from one neurons to another neurons and through those neurons to all the whole body. And here you can see this is the synapse. At synapse, they releases the neurotransmitter, and through neurotransmitters, neurotransmission takes place. And astrocytes and microglia are the supporting cells which helps neurons for better growth. Astrocytes are the cells which provide the neurotropic factors, which is very much required for neuronal growth. And microglia, these are the cells which are the phagocytic in nature. And whenever there is any kind of infection, so microglia, they clear that infection. So in case of Alzheimer's disease, accumulation of beta amyloid and tau protein, it takes place. So when tau hyperphosphorylation and its accumulation is there in the neurons, in that condition, microtubules of the neurons, they get destabilized and the neurodegeneration of those neurons takes place. Similarly, excessive accumulation of tau and amyloid beta also causes the activation of the microglia, which further exacerbate the neurodegeneration. So ultimately, it leads to the loss of synapse and neurotransmission loss in the brain, which ultimately leads to the pathogenesis and appearance of the different kind of symptoms associated with that disease. And amyloid beta is also been shown to disrupt the neurotransmission process. Not only it degenerate the neurons, but it also disrupts the neurotransmission process. And here you can see the healthy brain. This is the pictorial representation. And when symptoms get start, so this is kind of mild cognitive impairment when neurodegeneration is just started. And in case of advanced stage of Alzheimer disease, there is lots of degeneration in the brain. Symptoms I have already told you that learning and memory impairment is a major symptoms, and then ultimately it also lead to confusion with time or place. Then patient, they also have confusion in travel following conversation. So these are the major factors. And Alzheimer disease therapy mainly involve reduction of oxidative stress, several antioxidants being used for reducing the symptomatic relief, providing symptomatic relief. And then anticholinergic drugs are the major therapeutic approach. So what are other factors by which we can enhance the brain self-repair mechanism? Nowadays, approaches are being made to take patient-derived induced pluripotent stem cells. So first of all, scientists, they take fibroblasts, they convert them Alzheimer disease patient specific induced pluripotent stem cells. Then these induced pluripotent stem cells, they have the ability that they can differentiate into specific type of neurons, and these neurons, they mature, and these neurons, they can be used for drug discovery, and similarly they can be also used for transplantation into the brain. So there are two types of approach using stem cells to repair the brain damage. First one is exogenous and another one is endogenous. In exogenous approach, the neural stem cells isolated from the this induced pluripotent stem cells can be transplanted directly into the patient brain, while in endogenous approach, the stem cells which are already present in the brain of the patient can be targeted. They can be targeted to induce neurogenesis process or generation of new neurons. So these are the two approaches which are being used worldwide. So in exogenous approach, here you can see that induced pluripotent stem cell derived from the patient, they can be converted into the neural stem cells, and then these neural stem cells can be transplanted directly into the brain. And through endogenous neural stem cell targeting, we can enhance the neuronal regeneration process. So what is neural transplantation or regenerative medicine approach? So neural transplantation involve that dead or dying neurons replaced by implanting either fresh neurons or transplantation of neural stem cells. And after transplantation, these neural stem cells, they grow in the brain. But what is the limitation of neural transplantation approach? That transplant in the brain, there is hostile environment. So whenever we do the transplantation, in that condition, the transplanted stem cells or transplanted neurons, they die few weeks or few months post-transplantation. And then whatever functional recovery or symptoms recovery was there, so those symptoms again gets appear. So that's why this approach is not very much feasible. Similarly, there are several ethical and legal issues also involved in neural transplantation approach. Moreover, this is a very traumatic approach, and it cannot be used regularly for regenerative medicine. So how we can enhance the brain self-repair mechanism? So this process is known as neurogenesis. Neurogenesis is a formation of new neurons from multipotent neural stem cells, and this process plays a very important role in neurodevelopment. Similarly, now we know that neurogenesis takes place throughout life. Earlier, this concept was there that neurons formation takes place just once in whole life. But now we know that in the brain, there are stem cell population, and these neural stem cell population, they have the ability that they can proliferate, they can increase their number whenever there is requirement. And then after proliferation, they migrate at a specific site, and after that they differentiate into a specific type of neurons on the basis of requirement. and then they do the neurotransmission process. So in the brain, there are two main regions. Dr. Hiya Ghosh already told you about this. There are mainly two brain regions, hippocampus and subventricular zone, where neural stem cell population reside. And I tell you that there are several diseases where the population of endogenous neural stem cells gets decreased. Like in case of Alzheimer and Parkinson disease, the Parkinson and Alzheimer disease patients, they have less number of neural stem cell population as compared to the control subject. So that means this process is reduced in Alzheimer, Parkinson, and other neurodegenerative diseases. And this process can be targeted, and endogenous neural stem cell can be targeted to enhance the brain self-repair mechanism. So what is stem cell? Stem cell is the cell which has the ability to self-renew. They can make identical stem cells. They can maintain their pool. And whenever there is requirement, they have the ability to differentiate, and they can form the specialized cells. So they have the both self-renewal and differentiation ability. And neural stem cells has the ability to differentiate into specific type of progenitor cells, committed progenitor cells. Like neural progenitor cells, they convert into the neurons and interneurons, while glial progenitors, they convert into the oligodendrocyte and different types of astrocytes. So these are the specialized cells, and neural stem cells have the ability to differentiate into any type of neurons or astrocyte. So adult neurogenesis has the functional implication, and this process can be targeted to enhance the functional recovery in neurodegenerative diseases. So it can be targeted to reduce the symptoms in Alzheimer disease patient, and this is a very good approach for regenerative medicine in Alzheimer disease as well as in other neurodegenerative diseases. So here I would like to discuss a little bit about the drug repurposing. So our group is working on this aspect, and why drug repurposing is required. As you know that identification or making new therapeutic target takes lots of time, because target identification process is involved. Then you need to screen thousands molecule, and out of that 1,000 molecules, you screen identify few molecules, and then you do the lead optimization. And after that, you do absorption, dissociation, metabolism, excretion, toxicity study. And then it go into the phase 1, phase 2, phase 3 clinical trial. And after that, it go into the market. So ultimately, it takes 10 to 17 years times, even more. And then it takes lots of money, and it is also labor intensive. So in drug repurposing approach, what we do, that we take already available market drugs, which are being used for different diseases and which are already in the market. So in that approach, we do not need to do any kind of target identification, any kind of new screening of the molecule. Lead optimization is already there. So we just take the compound, and then we do the compound validation assay, whether it is useful in a particular type of disease or not. And then it can directly go into the different kind of clinical trials. So in our study, we used an innovative drug repurposing approach for Alzheimer disease. So this was a network project of the CSIR, where 5 CSIR laboratories participated in one project, CSIR, IGIB, CSIR, CCMB, IITR, ICT, and CDRI. So first of all, on the basis of behavioral and transcriptomic model, we have taken 2,200 known drugs which are already available in the market. These are the FDA approved 2,200 drugs which are being used for different diseases. And then on the basis of data mining and literature search, we identified few hits, and then we taken those hits in our Drosophila based model of locomotor activity reduction. So here we identified whether these molecules have any kind of neuroactive potential or not. And among 2,200 drugs or skin compound, we found 190 hits in Drosophila-based model of neurobehavioral deficits. So this work was carried out at CSR IGIV. And then out of these 190 hits, we identified that 20 fly hits we tested at the level of rodent neural stem cell proliferation and differentiation. And then we tested in rodent model of Alzheimer's disease. So here you can see that first of all, on the basis of data mining, we identified few neuroactive molecules, and then we screened those molecules in Drosophila preclinical model at CSR IGIV. And out of those, we identified few active molecules, and those molecules we have screened in stem cell proliferation and differentiation level in the culture. And then, so we have adopted multi-tier screening approach. First of all, data mining, then Drosophila-based preclinical model. Then we used neural stem cell proliferation differentiation model of the neural stem cells in vitro. And then whatever drugs were found positive on neural stem cell proliferation differentiation, those were studied in screening of potential CNS drug candidates in neurotoxin-based model of Alzheimer's disease. So this is the animal model of Alzheimer's disease. And then we have done the neurobehavioral neurochemical analysis. And ultimately, we identified the mechanism of action of finally emerged potential drugs. So here you can see the complete table. So these are the different drugs which are being used for different diseases like treatment of high blood pressure, diabetes, and so on. So we studied the effect of these drugs on neural stem cell proliferation, neuronal differentiation, and then in animal model of Alzheimer's disease, we studied learning and memory improvement. And here you can see that Ethos Oximite, this is a drug. It has the potential to enhance the stem cell proliferation, neuronal differentiation, and it also reverses the learning and memory impairment in Alzheimer's disease. So we have carried out the detailed studies using Ethos Oximite. So Ethos Oximite is an anti-epileptic drug which is being commonly used for the treatment of absence seizure in children. And how it works? It works mainly blocking the T-type gated calcium channel, and then it also reduces the continuous firing of the excitatory neurons. And one study showed that it also promotes neuronal differentiation in the red muscle-derived stem cells in vitro. However, whether Ethos Oximite has any effect on neural stem cells in vitro and in the rodent brain, it was not known. And how Ethos Oximite enhances the brain self-repair mechanism. So we have carried out studies to understand this aspect. So overall, our main aim was to understand how Ethos Oximite enhances the brain self-repair mechanism or in neurogenesis process. And then we also studied the mechanism involved. So this work was published in JBC. Ethos Oximite induces hippocampal neurogenesis and reverses cognitive deficit in an amyloid beta toxin-induced Alzheimer's disease rat model via PI3K, AKT, Wnt beta catenin pathway. So first of all, we have established stem cell culture. For that, we isolated neural stem cells from the rodent hippocampus region. And after that, we added different mitogenic factors like EGF and BFGF. So after addition of these mitogenic factors, neurosphere formation was there. So neurospheres are the bunch of thousands of neural stem cells, and these neural stem cells has the ability to self-renew themselves. And then we characterize these neural stem cells or neural stem cells or neurospheres using specific markers like BRDO, which is a marker for multipotent neural stem cell or proliferation marker. And then nestin is also marker of neural stem cell. And here you can see the colocalization. So that means these are the stem cells. After that, using specific type of factors, we differentiated neural stem cells into different kind of brain cells like neurons, astrocytes, oligodendrocyte, and then we characterized those brain cells using specific type of markers like neurons were characterized by beta tubulin, astrocyte was characterized by GFAP, And here you can see the colocalization of beta tubulin and GFAP. So both type of cells are present in culture. And then oligodendrocytes were characterized by CNPH and myelin basic protein. So here main aim was to ascertain, yes, whatever we are culturing, those are the stem cell culture only, and we have the very established protocol of stem cell differentiation. Then after that, we treated stem cell culture, neural stem cell culture with Ethos Oximite, different concentration of Ethos Oximite. And here you can see that at 100 micromolar concentration, Ethos Oximite was able to enhance the stem cell proliferation. Similarly, at 50 micromolar also stem cell proliferation was high, but at higher concentration, Ethos Oximite reduces the stem cell proliferation. So it is also possible that at higher concentration, it is causing the toxicity to the neural stem cell. And then here you can see BRDO positive neural stem cell was significantly higher in Ethos Oximite treated neural stem cell culture. So that means Ethos Oximite enhances stem cell proliferation of neural stem cells in vitro. And then we studied the effect of Ethos Oximite on multipotent neural stem cells. And here you can see that we studied the number of neurosphere as well as their size, because number of neurosphere and their size is a direct indicator of multipotency of the neural stem cells. And here you can see that the number of neurosphere as well as their size was significantly enhanced by Ethos Oximite in neural stem cell culture. Similarly, here you can see the colocalization of nestin with BRDU. It also suggests that the colocalization of nestin and BRDU in Ethos Oximite treated culture was significantly upregulated, which suggests that Ethos Oximite enhances the self-renewal of multipotent neural stem cells. As I said that after stem cell proliferation, they have the ability that they can differentiate into a specific type of neurons. So for that, we studied effect of Ethos Oximite on neural stem cell proliferation as well as their differentiation in the brain. So adult brain, they were treated with Ethos Oximite, and after a specific time duration, we dissected out the brain, we cut the brain sections, and then we did the immunohistochemistry using specific primary antibodies for specific stem cell proliferation marker. Here we have used BRDU, and here you can see the small dots. Those are the neural stem cells. This is the higher magnification images. Here you can see the small dots. So these are the neural stem cells which are present into the hippocampus region. So in case of Ethos Oximite treatment, it increases the stem cell number in the hippocampus region of the brain. And similarly, it also enhances the stem cell number into the subventricular zone. So this is the subventricular zone. Here you can see subventricular zone, and this is the stratum lining. And in subventricular zone also, it increases the stem cell proliferation. And then it was further established using phosphohistrone S3, which is also a mitogenic factor. So this suggests that not only Ethos Oximite enhances stem cell proliferation in vitro, but it also increases the stem cell proliferation in the rat brain. And after that, we made animal model of Alzheimer disease by directly injecting amyloid beta 1 to 42 fragments into the brain, into the hippocampus region of the rat. Here you can see the histotoxic instrument, and then we make a small incision on the skin, and then we, through histotoxic coordinates, we inject amyloid beta directly into the hippocampus, and then we stitch the skin. And after 3 weeks post injection of amyloid beta, we do the neurobehavioral analysis in order to see the effect of amyloid beta on learning and memory ability. When there is significant reduction of learning and memory ability, then we make sure, yes, these animal model of Alzheimer's disease are now prepared. So after that, what we did, we treated those Alzheimer's disease animal model with Ethos Oximite, and here you can see that amyloid beta significantly inhibits the neural stem cell proliferation into the hippocampus region of the brain. But when animals, they were treated with Ethos Oximite, in that condition, amyloid beta mediated inhibitory effects on stem cell population was reversed by Ethos Oximite. So Ethos Oximite activates neural stem cell proliferation in the hippocampus region of rat model of Alzheimer disease. And then we studied its effect on neuronal differentiation and its maturation. So in order to study neuronal differentiation, we used a marker of double cortin. For that, we did the co-leveling of BRDU and double cortin. As I said, that BRDU is a marker of proliferating stem cells, while double cortin is a marker of newborn neurons. So the yellow shows that proliferating cells, they are differentiating into the neurons, while only green, it suggests that these are the proliferating cells, while red, it suggests that they are the newborn neurons, and yellow is the neurons which are being converted, neural stem cell being converted into the newborn neurons. And here also you can see this is the quantification of newborn neurons into the hippocampus region. And here you can see that Ethos Oximite significantly upregulated the BRDU double cortin positive cells, which suggests that the number of newborn neurons is significantly high in Ethos Oximite treated animals. Similarly, the amyloid beta mediated inhibitory effects on newborn neuron population was also protected by the Ethos Oximite. And then we did the maturation analysis of the neurons, and we found that yes, whatever neurons formation was there, those neurons being converted into the mature neurons. So ultimately, it enhances the neuronal differentiation and also their maturation into the hippocampus region. After that, we studied the mRNA expression of several neurogenic genes, because neurogenesis is a very complex process, and it is regulated by hundreds of transcription factors, several genes, co-activators. So we studied the effect on some critical neurogenic genes and critical transcription factors. And we found that Ethos Oximite significantly enhances the expression of those neurogenic genes, and it also protects amyloid beta mediated reduced expression of those neurogenic genes. And then through in silico analysis, we identified possible targets of Ethos Oximite. We did the in silico docking studies with several proteins or several molecules which are involved in regulation of neurogenesis process. Out of around 200 molecules we screened, or possible target we screened, we found AKT1, DKK1, and GSK3 beta. These three, they have the ability that where Ethos Oximite can go and it can bind, and it has very good C-Docker energy. And then now we know that Wnt beta catenin signaling is a very important signaling pathway which regulates the neurogenesis process, and Wnt beta catenin signaling is also gets reduced in Alzheimer disease, and it is also regulates the neurogenesis process. So ultimately, this can be also a therapeutic target. So here we hypothesize whether Ethos Oximite has any effect on this signaling or not. And then Wnt signaling is also required for long-term memory formation, and Wnt knockout animals, they have the reduced brain growth. So this process is very much involved, Wnt signaling is very much involved in regulation of adult hippocampus neurogenesis. So this is the Wnt pathway. I will not go in detail. I will just present the data, whatever we got. So first of all, we studied the expression of different genes, different regulators of Wnt beta catenin pathway. So in Wnt beta catenin pathway, there are several negative regulators like Wnt inhibitory factors, DKK1. So these factors, they inhibit this pathway. And what we found that Ethos Oximite has a negative effect on these regulators, and it reduces the expression of these negative regulators. Ultimately, it enhances the Wnt beta catenin pathway. Similarly, the expression of different receptors of this pathway was enhanced by the Ethos Oximite in amyloid beta induced model of Alzheimer disease. So ultimately, it activates the Wnt beta catenin pathway by inhibiting the negative regulators, by enhancing the receptors expression, and then by reducing the expression of different degradation complex molecules. So ultimately it activates the Wnt beta catenin pathway into the hippocampus region. Similarly, on the protein level also we found that by phosphorylation of Wnt beta catenin pathway, it activates this pathway, and ultimately it enhances the hippocampal neurogenesis. And when we blocked this pathway using specific inhibitors, like for this we used DKK1. DKK1 is an inhibitor of Wnt beta catenin pathway, and when we use DKK1, in that condition, here you can see that stem cell proliferation was completely diminished. Like green fluorescent neural stem cells are very less in number as compared to the control. And in Ethos Oximite treated culture, the green fluorescent neural stem cell is significantly higher. But when we treated co-cultured with DKK1 and Ethos Oximite, in that condition, neural stem cell population was there. So ultimately, when we blocked this pathway, in that condition, Ethos Oximite mediated effect on neural stem cell population was significantly blocked. Similarly, on neuronal differentiation also we got the similar result, and Ethos Oximite mediated effects was blocked by Wnt beta catenin pathway. And here we did the very interesting experiment. What we did that we directly injected Wnt beta catenin pathway shRNA, that is Wnt beta catenin siRNA, directly into the hippocampus region. We injected that siRNA into the hippocampus region, and then we studied co-localization of BRDO and Neuron in order to see the effect on neural stem cell population as well as their neuronal differentiation. And here you can see that in Ethos Oximite treated animals, the number of BRDO and Neuron positive cells was significantly upregulated. But when we injected beta catenin into the brain, in that condition, the process of new generation of the neurons was significantly reduced. Similarly, stem cell proliferation was also reduced. And the protective effect of Ethos Oximite through this pathway was also blocked by beta catenin siRNA. So ultimately, we can say that yes, this pathway is involved in regulation of hippocampal neurogenesis by Ethos Oximite. And this pathway is also intermediate PI3K AKT pathway. It is also intermediate in Wnt beta catenin pathway. And here also we can say that Ethos Oximite activates PI3K AKT pathway into the hippocampus region of the rat brain. And similarly, when we inhibited this pathway using specific inhibitors, that is LY294002, in that condition also, here you can see the TUJ1 positive neurons, their number was significantly reduced by inhibition of PI3K AKT pathway. So we can say that PI3K AKT Wnt beta catenin pathways are involved in regulation of hippocampal neurogenesis, and Ethos Oximite plays its protective role through these pathways. And ultimately, we studied effect of Ethos Oximite on hippocampal neurogenesis, because when stem cell proliferation is there, they are being differentiated into the neurons, and then they are also being converted into the mature neurons, and it is being happening through Wnt beta catenin pathway. Then we studied effect on learning and memory ability in Alzheimer disease rat model, and here you can see that Ethos Oximite per se has no effect on learning and memory ability, but amyloid beta significantly reduced the learning and memory ability in animal. And when we treated those animals with Ethos Oximite, learning and memory ability was significantly upregulated as compared to the amyloid beta. So this suggests that learning and memory ability was enhanced by Ethos Oximite. And this is the schematic representation of mechanism of action. And here you can see that Wnt beta catenin pathway was involved in regulation of hippocampal neurogenesis through Ethos Oximite, and Ethos Oximite enhances hippocampal neurogenesis. It also protects and increases the cell survival, neuronal survival into the hippocampus region. So overall, we can summarize that through our drug repurposing approach, we identified a molecule which has the ability that it can enhance the brain self-repair mechanism through targeting endogenous neural stem cell population into the hippocampus, and it increases the hippocampal neurogenesis through activation of Wnt beta-catenin pathway, and ultimately it recovers cognitive deficits in rat model of Alzheimer's disease-like phenotype. So I would like to acknowledge my students, Dr. Sasikant Tiwari, Dr. Swati Agrawal. Dr. Sasikant Tiwari, he is University of California, San Diego. Dr. Swati Agrawal, she is in John Hopkins. Dr. Bresket Set is also faculty. And Dr. Anuradha Yadav, she is in TIFR. And my collaborator, Dr. Salin Gupta. And then funding I received from CSIR, DBT, DST, and Lady Tata Memorial Trust, Young Scientist Grant. Thank you so much for your kind patience. And I'm open to take any question. Thank you.
Thank you, Dr. Rajneesh. Does anyone have any questions? Okay, so I have a like very basic question. Like we have seen amyloid beta hypothesis almost there from almost 20 years. And maybe I wrote some papers around 2 years back that some of the potential drug failed in because of this hypothesis. What are your views on this particular aspect?
Yes, you are right that only amyloid beta hypothesis is not involved. Earlier, it was thought that only accumulation of amyloid beta accumulation is responsible for pathogenesis of Alzheimer's disease. Now we know that tau hyperphosphorylation is there. Now we know that cholesterol hypothesis is there. So multifactorial approach we can adopt. So that's why it is now required that we identified so many targets, and then we work on multifactorial approach. Then only therapy of Alzheimer's disease can come in market. If we target only Alzheimer's disease hypothesis, this amyloid beta hypothesis, it will not make any sense.
Okay, thank you. And like I saw very interesting data from Ethos Oximite. I just wonder that what was your initial criteria for identifying Ethos Oximite, because you have shown some 2000 transcriptome that you have isolated, and then what was the main pathways you focus to find out these hits?
Yes. Yeah. So first of all, as I said that first of all, we took 2200 FDA approved drugs, which are already available in the market. Then on the basis of literature and data mining, because literature is available. So on the basis of data mining, we identified few molecules which has the neuroactive potential. And then we did the transcriptomic analysis. And in transcriptomic analysis, we mainly focused on specific pathways like Wnt beta-catenin pathways, TGF-beta, Notch, sonic hedgehog. So we identified around 20 pathways, which mainly plays an important role in regulation of neurodevelopment, transcription factors, regulation, then neurotropin signaling. So we identified around 20 cell signaling factors. And on the basis of that, we took those molecules into the Drosophila-based preclinical model. So this model is a learning and memory deficit model. And then they also have the hypolocomotion, they have reduced locomotion. And then those identified hits, we screened in Drosophila-based model. And then whatever neuroactive molecules we identified in Drosophila, we took them in neural stem cell proliferation and differentiation level. And then we identified that, yes, these molecules, they have the ability to enhance neural stem cell proliferation and differentiation. Then we tested that in animal model of Alzheimer's disease. And in CSR, CCMB, we screened them for Parkinson's disease. In CDRI, we screened them for stress. So it was a CSR network project. So at IITR, we screened them for Alzheimer's disease.
Okay, thank you so much. And I hope that Ethos Oximite could be utilized for potential molecule in Alzheimer's disease. And it could be a great contribution from CSR lab. Thank you so much. And I would like to invite Dr. Palla for vote of thanks. So thank you so much for your exciting talks. Yeah.
Thank you.
Thank you, Dr. Rajneesh, for accepting our invitation and for all your precious time. It was a great opportunity for us to hear from you. And we really look forward to meet you in person. And we invite you in NIPER-Ahmedabad once we are out of this pandemic. And thank you very much. It was really very inspirational. And we look forward for more collaborative work. And hopefully your drug, Ethos Oximite, may evolve as one of the molecules that can be used for treating Alzheimer's disease. Thank you very much, Dr. Chaturbaidi.
Thank you so much. Thank you, everyone. Thank you. Namaskar.